Hey guys, my name is Khashan, and we'll talk about the CrudeDrugs drug development process, the receptor-ligand interaction, and the structure-activity relationship. As you go through the 20 pages of notes, I want you to focus on these reading objectives. These are the objectives that I expect you to know before the class, so you can take your IRAT and TRAT, and then we will have team-based applications. So the objectives are the following. You should be able to describe the steps involved in each stage of the process of the drug development. That is drug discovery, drug design, and drug development stages. And then make sure you are able to compare between the in vitro, in vivo, and in silico experiments, and list different types of resources for lead discovery. Okay, and define the target selectivity and the relationship between target selectivity and the undesired side effect. Define combinatorial therapy and explain why it can be advantageous. Identify advantages and disadvantages of finding lead compound using natural products. Explain the method used in identifying the structure-activity relationship and pharmacophore. Differentiate between pharmaceutical, pharmacodynamic, and pharmacokinetic phases. Discuss the advantages and disadvantages of using herbal medicine instead of prescription drugs. Appreciate or appraise the risk of drug interaction with herbal medicine and the role of pharmacists to prevent it. And then make sure you become familiar with the types of the drug targets, the enzymes and the receptors with examples. Understand the chemical nature of proteins, just the general overview in the video. And understand the process of how the the ligand, which is a drug, which could be a drug, binds to the receptor, and the interaction, that type of interactions that forms, and the induced fit or conformational change or structural change that occur to the active site, which results into the signal transduction, and then in case of the receptor, the ligand leave the receptor after the signal transduction. Then differentiate between the binding site of an endogenous ligand and the allosteric binding site. Make sure you know what's the definition of allosteric binding site. Differentiate between types of receptor-ligand intermolecular bonds, rank them by strength, and relate this strength to the affinity of a ligand, and as well as the KD constant. So KD is actually a reflection of the affinity. And then relate the impact of these bonds on the induced fit and the biological effect. Relate the structural activity to the drug target interaction. So how the structures, part of the structures, are relating to the activity, and how that plays a role in the producing the effect. Basically, basically the interactions between drug and the target leads to the activity. Without these interactions, you will not have activity. And then differentiate between 3D pharmacophore, generalized bond pharmacophore, understand the similarity principle and how it relates to identifying the pharmacophore. Differentiate between isosteres and bioisosteres and what's their importance in the drug design. And then appreciate the importance of stereochemistry and the impact it has on the pharmacological activity of drugs. As far as the function groups that you will see, if you cannot remember them, that's fine. Just make sure you get familiar with them. So we will not memorize any of these structure groups. Just try to be familiar with them. Let's talk about CrudeDrugs and pharmacognosy. First of all, a crude drug is any naturally occurring unrefined substance derived from an organic or inorganic source, such as a plant, animal, bacteria, organs, or a whole organism, for the purpose of either diagnosis, cure, mitigation, treatment, or prevention of a human disease or an animal disease. And then pharmacognosy is basically the knowledge of the drug, basically the study of these crude drugs, which involve the study of the physical, chemical, biochemical, and biological properties of the drug, the drug substances or potential drugs that are from natural origin, as well as the discovery of new drugs from natural sources. Okay. Let's start with the drug discovery from natural products. The first thing that comes to our mind when we talk about the drug discovery from natural products is that with the presence of the advanced and modern techniques of the synthesizing new molecules and with the computational methods, that you think that the natural products are no longer significant, no longer play a significant role in drug discovery. And indeed, for the past 2 decades, there has been a decreased emphasis on screening and searching um, natural products to look for new drugs or lead compounds. Yet, in a review article recently published, you can find it in this reference book number 3. The article states that between the year 1981 to 2006, for the total number of 1,184 newly discovered drugs, 5% of them were actually unmodified natural products, and 23% were semi-synthetic agents based on natural products, and 14% were designed based on knowledge of natural products. So this is a total of about 28 and 14, so that's like 40, close to 40% of the newly discovered drugs still being impacted by the natural products. So the launch of new drugs from natural products still continue to play a role in the 21st century. Okay. Now, why is that? Why is natural products still playing a role in the discovery of new drugs? The reason is that studies show that natural products or libraries that have a collection of substances derived from natural products are more drug-like. They have more drug-like properties. When we talk about drug-like properties, we talk about the drugs currently in the market which have good oral availability. They can be taken orally, they have good distribution, etc. All these features we explained earlier, they have less side effects. So these natural products, they have more drug-like properties, and this is expected of course because they are produced by living organisms. So the products that are produced are similar to the chemicals that available already in our bodies, and then so it's more like a drug. It has a drug likeness property. So this is one reason. Another reason is that why we should consider natural products as a source for discovering new drugs is that natural products actually occupy a different region in the chemistry space. And what I mean by chemistry space is so if you have a database of molecules, then the natural products would actually occupy a certain region in space, in the chemistry space, that is different from the synthetic compounds, and I'll explain this in a minute in further clarify it with what I mean by chemistry space. But what's important is when you design, when you look for discovering new drugs, you wanna look to as much diverse as molecules. You wanna look at the most diverse molecules, number of molecules that are possible, so you could actually explore all the options you have. So the synthetic compounds would be this similar um, from the natural products, and then you would basically want to include the natural products to make sure you cover all the possibilities. Okay, and then so many of the natural compounds have totally novel structures. So basically, the natural products will have structures that are totally different from those synthesized. So if you ignore these natural products, then um, you are basically decreasing the chance of finding chemicals that can be active. Let me explain to you what I mean by chemical space. So if this is a two-dimensional space, okay, and the synthetic molecules are gonna fill, for example, this region of the space, and because they are similar to each other, are gonna fill, let's say, this region of space, and then the natural products are gonna fill, for example, this region of space. Okay, and so your goal is to include both of these databases such that you increase the chance of finding a drug. Okay. Now I want you to think about these coordinates and consider what could be on these coordinates, and can we have a third maybe dimension, or can we have more than one, I mean three coordinates to describe the chemical space? Try to think about that. Okay. Okay, so we have explained why the discovery of lead compounds or new molecules from natural products is still an advantage. So what would be the disadvantages of looking at natural products and use them to find lead identification? First, natural products extracts are actually considered incompatible with the modern rapid screening techniques in use. So the in vitro or in vivo techniques used by pharmaceutical industries, they are not compatible with the natural product extracts, so this can cause some problems. Second, the isolation and purification of the natural products and the active principles is very time consuming and costly, and so this can delay the process of the discovery. And also natural products may only be present in small quantity, and so this restricts its availability. Um, so in order to perform these tests, you probably need to synthesize more of these products. And what's the problem with the synthesis is the fact that these natural products are complex, and then the synthesis for these complex structures is impractical and could be expensive and would consume time. So these are the disadvantages for using natural products as a source for a lead compound. Okay, I should also notify you um, that you should never think or um, um, basically assume that within all these years of searching for drugs that we have explored all the available natural products. In fact, if you only look at the plants, we have only um, explored 20% of the available plants, and so there is still a lot of resources and natural products that we could use to look for lead compounds. And notice here I'm only referring to plants. There is also animals and the neurotransmitters and hormones. These resources which are part of the natural products. Okay. Okay. So far we have described how natural products can be useful in discovering lead compounds. What about the herbal medicine itself? Are they better than using the prescription drugs or not? Um, there is no simple answer to this, but there are certain factors we need to consider when we look at this. So, first of all, the, um, the World Health Organization um, states that about 80% of the world's population uses herbal medicine as part of their normal health routine, which means the herbal medicine is very is used very is used commonly and very often. So, and the reason for that is that people think or believe that natural products are inherently better than synthetic drugs because they think it's comes from nature, so it's natural, it should be healthy and better than drugs. Besides, the herbal ads always convey the message or the attitude that herbal medicine is safe and effective. Um, in addition, the herbal medicine is far less expensive than prescription drugs. So for these reasons, the herbal medicine is used or is popular in the world. Okay. However, if you think about it, herb, the herbal medicine are basically drugs because they have chemicals, and no matter how many times the label on that product says the word nature, frankly, these herb, these herbs contain chemicals, they have pharmacological activity, they can affect the person's health, and they are not pure substances. In fact, they are far away from pure substances. They are a complex of many substances, as we'll explain shortly. And they, on top of that, they can interact with many prescription drugs. So these herbal drugs are good as source for designing drugs and for discovering lead compounds, but by themselves, these herbs are not comparable to prescription drugs. Okay, the active principle, which is the active component in the herbal drug, also normally is present in small amount. So the herbal medicine um, that may be expected to have less activity than a pure drug. Okay. Herbal medicine also contains large variety of different compounds. So some of them could be active, others are not. Some of them could be toxic. So this is something, a factor you want to keep in mind when you compare, when you think about the use of herbal drugs versus prescription drugs. Okay. Another factor to keep in mind is the fact that a typical herbal medicine may contain dozens of different compounds, and in many of these cases we simply don't know which one of these components are responsible for the pharmacological activity. In fact, sometimes these components work synergistically, which means they help each other, they add the pharmacological effect to each other to produce the desired pharmacological effect. And when you try to separate each component by itself, it has no pharmacological effect. So the pharmacological effect will be lost from this herbal. We will discuss an example like that very soon in details and show how an herb can have many chemicals and some of them could be active, others inactive, and then when combined together you might see the pharmacological effect. And it's only in rare cases when you have the active ingredient of an herb is isolated and characterized and tested for activity. Normally it's the combination of this with other drugs. I want you to try to look for the definition of the synergism. There is three terms that a drug, that two drugs can the three ways that two drugs can interact with each other. One is through synergism, is when you have an active drug and then another active drug. They both synergize and activate each other and enhance the activity of each other. There's the second way is called potentiation, where you have one drug inactive, the other one is active, and then the inactive drug increases the activity of the active drug. And the third way is called addition, which is from its name. If you have an active drug and another active drug, when you combine them together, then the activity would be simply the addition, the total activity for these two drugs. And unlike synergism, in the case of synergism, you will have the activity multiplied by having two active drugs. Anyway, so look for the terms synergism, addition, and potentiation just for your own, for your own education. We're gonna visit this again. Okay, it's the three terms. Next is that the herbal preparations are most often used as crude mixtures and not standardized. Okay, so this is one factor you want to consider, that these herbal medications are not standardized or analyzed um, for their contents. So you might have different mixtures from one manufacturer and then from and another totally different mixture from another manufacturer. The amounts, the way these drugs are analyzed, all this will affect the, pharmacology of these, the, the pharmacological effect of these mixtures. Okay, so so again, they are not standardized. In addition, um, their action and interactions and side effects, because they are not standardized, you cannot predict their action or side effects or interaction. Okay, and then there is a note here that I want you to pay attention to, which is I want you to be aware that it is essential that we, for herbal medicine to be analyzed and for the herbal component to be studied to find which one has the active principle. But in many cases, we are not able to do that. So, and because of that, we go back to the use of prescription drugs. Okay. Another factor to consider is that some plant extracts have a wide variety of different active principles that act together to produce the beneficial effects. In other words, you might have, in this case, we provide an example of the aloe plant, which is also referred to as the wand of heaven. It's a cactus-like plant found in the deserts of Africa and Arizona, and it actually has many pharmacological uses, but it also includes many components, many chemical structures that are responsible for these effects. And it's not just one of them, again, that's responsible for these effects, but the combination of these all together. So you will have some anti-inflammatory, analgesic, antimicrobial molecules or extracts within this plant that are responsible for these actions. However, um, the clinical efficacy for these is still um, you know, not significant. So, and another example is the ginseng, for example, which is believed to enhance the concentration, stamina, alertness, and ability to work. However, clinical studies comparing ginseng with placebo show that there is no statistical significance for the ginseng. And placebo basically means is when you take something that does not have any pharmacological effect. I'll give you an example. Let's say that you give a patient like a candy, and then you tell them that this is a medicine, so they get better by taking the candy, thinking that it's a medicine. So it's like a psychological effect, when in fact there is no any medicine in that candy. So, and then I will show this video here of how a drug is being synthesized. This is from the traditional folklore, from like some old um, ways of treating epilepsy. It's just a funny way to give you the message about the placebo effect. Okay. Okay, so this is how the drug is synthesized. Um, you heat some water and then you add some juicy spiders, and you get a pedigree dog, and then use the dog turd on top of that. This is supposed to be a cure for epilepsy, and then you simmer for about 20 minutes. So this shows that an example of what a placebo effect is like. Okay, so basically some of these herbal drugs have no pharmacological effect, but just by the effect of placebo. Okay, and then I selected one example and discussed the medicinal chemistry for that herb in details, and then you could look for the similar examples in the supplemental material to see the medicinal chemistry for other plants. The one I selected was the echinacea. I'm not sure if I'm pronouncing this right or not, but it doesn't matter. It's an immunostimulant that is used to lessen the symptoms and duration of cold and flu. Okay, and it's also used sometimes as a wound healer, which makes sense. If it's an immunostimulant, then it improves your immunity and then it should reduce the symptoms of cold and flu. Right? Then the studies shows there is no doubt that echinacea has immunomodulating properties. However, we are not sure which ingredient is responsible, or actually there is no single ingredient that is responsible for this feature. So as you will see, it's a combination of many ingredients that are responsible for this activity. Okay. Besides, there's a problem with the standardization for preparing such an herb. First, there are 3 kinds of species available, which is the Echinacea purpurea and Echinacea angustifolia and Echinacea pallida. So some of these preparations have their active compounds in the roots, other have them in the aerials, other have them in the whole plant. So again, by selecting which plants and selecting which part of the plant, besides how these preparations are extracted, all this can affect the composition of the material that we are using. So besides, the reproducibility of such material will be not possible. So again, standardization is an issue. And then I mentioned just few components I selected that has some activity. For example, the 2 polysaccharides, and I'm not gonna read these 2 names, PS1 and PS2, were found to be immunostimulatory. Okay, so they basically stimulate the phagocytosis and blah blah blah, stimulate the macrophages, causing enhance the immunity. And then there are 3 glycopeptides that were separated and found to stimulate the beta cells as well. And there's some other effects like besides the immunostimulants property, and there's some anti-inflammatory effects, and these are some of these structures that were extracted. I do not expect you to memorize any of these structures or to memorize any of these functions. This is just an example of how an herb would have many components and it's difficult to standardize it. Second, these components, some of them are active, others are not, and some of them have the activity, the immunostimulant activity, and it's the combination of these activity that causes the synergistic effect, the pharmacological effect. Okay, and you will see many of other herbs, the medicinal chemistry of other herbs, and this reference here, which I also posted in on Blackboard. Okay, so this is for your own information. So finally, I want to talk about an important thing, which is the drug interaction of these herbs. The most common drug interaction of the herbal medicine is the blood thinning effect. So anytime you think of herbal medicine, you should consider that these medicines should not be combined with warfarin, aspirin, ibuprofen, because they will have anticoagulant effect, and then this effect will be potentiated, and then we will see side effects of the patient will be bleeding. And then other effects are like protease inhibitors. So basically these herbs can have the protease inhibition effect, the cardiac glycosides effect, and the immunosuppressant effect of the cyclosporine. So these are all possible interactions. And another example of an interaction is, for example, the salt Jones wort, which is an herb. It's used to treat depression and anxiety, and it's believed to act through by inhibiting an enzyme called monoamine oxidase. So this product, for example, should not be combined with another monoamine oxidase inhibitor, because this might potentiate the side effects. Okay, let's talk about the drug development process, which goes through the discovery of the drug, modifying the drug or optimizing the drug, and then the development of the drug, which is basically manufacturing and preclinical trials. So from the first stage of finding the lead, which has 6 different steps, starting with choosing the disease, choosing the drug target for that disease, and then establishing the testing procedures that are appropriate for the drug target and the disease, and then finding a lead. So a lead compound means it's a new molecule that's never been used before, and then isolation and purification of that lead compound in case it's like part of a complex of compounds, as in the natural products, for example, and then determining the structure for that lead if we are not sure yet what that structure is, and then we optimize this lead, basically optimize the pharmacokinetic and pharmacodynamic and pharmaceutical properties of the lead compound. So there are actually 3 major areas where we could optimize this lead: pharmaceutical properties, meaning their solubility, their dosage forms, formulations, the release from the tablets, etc., and then the pharmacokinetic properties, which is how the body affects the drug, how the drug is absorbed and then metabolized and excreted, and then also optimize the pharmacodynamic properties, which is how the drug binds to the molecule. Before these, we identify the relationship between the structure of that molecule and its activity, and we identify the pharmacophore. So once we define these, then we could optimize the other features. The last stage is the clinical and preclinical trial, and then manufacturing the drug, patenting it, and registering the drug, marketing the drug, and then making money. The first phase is called the drug discovery, which could be described as the phase of finding the lead compound. The first step in that phase is choosing a disease. For a pharmaceutical industry to choose a disease, they want to make sure that the money they receive from selling the newly developed drug outweigh the cost of this drug. The drug development process is very expensive, and for a pharmaceutical company, they want to make sure that they are able to make profits out of this new drug. And to do that, there are some questions that they look at before they choose which disease they want to work on. The first question is whether the disease is widespread or not. So diseases like cardiovascular, ulcers, malaria, etc. Is the disease widespread? Compare, for example, malaria to cardiovascular, or compare a rare disease to diabetes disease, which is very common. So you want to make sure that the disease is widespread, so you could have as many patients taking the medication as possible. And then, is the disease affecting the first world or the developing world? Obviously, the ability of the patients in the first world to purchase the medication would be higher than in the third world countries or the developing countries. So the pharmaceutical companies are more concerned into designing drugs to treat diseases that are spread into the first world. And then also you want to look at the advantages and disadvantages of the drugs already available in the market. Well, first, are there drugs already in the market? If so, what are the advantages and disadvantages of these drugs? Sometimes minor improvements in the drug available in the market could actually make the new drug very profitable. For example, if you have a drug that's taken twice a day and then you discover another one that can only be taken once a day, so you extend the duration of action for that drug, then this would be a big step in advancing the treatment for that disease. And then finally, is there a market advantage for that disease from the new therapy? So the new drug that you are planning to discover, is it gonna have advantage over the other drugs, and what advantage is that? Okay, so by answering these questions, a pharmaceutical company can decide on the disease they want to work on finding a new drug for. The second step in the drug discovery phase is choosing the drug target. So after you have chosen the disease, you need to identify the target for the drug you're trying to design. Okay. Normally, in order to have a pharmacological effect for a drug, there must be a target for that drug. And understanding the biomolecules involved in that disease would help you identify that target. For example, if you know that there's an enzyme or receptor involved in the in the disease that you are interested in, then you could actually design a drug to either block that receptor enzyme or maybe enhance the receptor or enzyme. So normally the function of the drug is either blocking the receptor or enzyme or enhancing it. When it blocks that enzyme or receptor, we call it antagonist, and when it enhances it, we call it agonist. We will visit these again later on in later sessions actually. An important concept here when you design a drug for a target is what's called target selectivity, which means when you design a drug, you want to make sure that it selectively binds to the specific target you want it to bind to. You want to avoid having a drug that binds to other targets. This is important because you could minimize the side effects that can result from the drug binding to other targets. Okay. This concept can also be referred to as finding the magic or silver bullet. So you can imagine having a magic bullet that would attack only certain receptors or certain targets but avoid the others. And the more selective your drug is, then the less side effects you will have for that drug. I want you to consider another term called target specificity. Try to look up the difference between target selectivity and specificity. Okay. And then I also want to give you a caution here when you look for drug targets. So when you define some targets for a disease, you might see more than one target, or let's say that you find one target for a disease, and you might think by binding to this target, inhibiting it, or enhancing it, then that should solve the problem. But our body is very complex, so by blocking one enzyme or receptor, the body sometimes is able to find other routes around it. And in this figure here, we illustrate this concept using a simple example where we have commuters trying to get to the downtown, and this is the main street. Okay, and then if you block the main street, then the commuters will try to find other ways to reach the downtown, like here. The same could happen with our body. So you might block one receptor or enzyme, and then the body will be able to find other ways to reach to the to continue acting, causing the disease. So the solution for this would be combining more than one drug and targeting more than one target. In this case, we use what's called a combination therapy. And the advantage of this actually is not only to control the disease, for example, but also you could lower the amount of drugs that you are using, that you are combining together, such that you can still achieve the same effect, but you would have less side effects from these drugs. We will visit this concept again. Okay. Now the next step in the phase discovery and the drug discovery phase is establishing testing procedures. Okay, so in this step here, we will work on designing experiments or tests, and this step actually is related to the one before it, which is choosing a drug target. Because let's say that you identified a receptor or enzyme, but then you want to test that you want to test the action of this receptor or enzyme, or the effect of binding to this receptor or enzyme on the progression of the disease, for example. So then you want to design certain experiments or testing procedures for this target enzyme, and check what drugs could actually achieve the goal you want without enhancing or blocking that target. And at this stage, it would be too early to, for example, test the drug on human. So we use other testing testing procedures. There are three in this case, which is the first one is called in vivo testing procedure, and in vivo means within the living. So you could test the drug on a living organism like animal or plants. Actually, it can also include human, but like we said, at this stage we do not use the human. And then there is the in vitro, which means within the glass. And in this type of testing, we use test tubes, which contains, for example, isolated cells, tissues, enzymes, receptors, like our target receptors, and then we see whether the drug binds to it or not, how it activates it, etc. And this step is much cheaper and easier to perform than the one before it. Okay, so the in vitro is actually less expensive than the in vivo, it's cheaper. And it can also be automated, such that you could perform a large number of tests within a short period of time. And this process of automation is called high throughput screening, HTS. So you could, for example, have 1,000 compounds tested all at the same time simultaneously, using on 1,000 test tubes, and the process could be automated. You could test all thousands of compounds within short time, within a few days. Okay. The third type of testing procedure is called in silico, which is my favorite. And in silico stands for in silicon, which is basically the material used to make up the computer chips. So in silico basically means that you are using computer programs to perform the test. So you use your fundamental knowledge in chemistry, biology, and you design software to test whether some drugs are going to be active or not. And these kind of tests or screening are called virtual screening. And what's interesting about these tests is that they are even less expensive than in vitro tests. So in the case of in vitro, you're gonna need time and test tubes, etc. But in silico, once you have a computer and then the software, then you could use it as many as you want, and it's typically the cheapest among all these three. And that's why it is the first one that's used, because if a drug is gonna fail, you would rather have it fail when it's still cheap, when it's not costing you a lot of experiments, and then you move to the next one in vitro, and then you move to the in vivo, and then as we will see later on in later stages of the drug development, you could do the clinical tests. Okay, so back to here, the in silico is the first one that is used because it helps you eliminate drugs that are unlikely to be active. Okay, and this is just for your information. There are other techniques that could be used to screen or do the tests, such as NMR spectroscopy, surface plasmon resonance, scintillation proximity assay. I'm not sure what that is. and isothermal titration. Anyway, so this is just for your own reference. If you are interested in reading about them, you could go to this reference here, number 4. Okay, but these are the major ones I'm interested in. Okay, so we are still at the drug discovery phase. We have defined the disease, we defined the target we want to look at, and we define the testing experiments we're going to use to check these targets. And then the fourth step is finding a lead compound. So first of all, a lead compound is simply the first compound that is discovered for a certain target. It's actually used as the starting point for the design of the drug. I'll give you an example. So penicillin, for example, penicillin G, which was discovered by Alexander Fleming, it was the first compound that led to the discovery of all the penicillins and cephalosporins antibiotic. Okay, so once you find a lead compound, a starting point, then you could actually use it to design many other drugs that have the same mechanism of action. Okay, so our goal here is to look for that lead compound, a pioneer compound that would actually open up the door for designing new drugs. Okay, and because the lead compound is the first drug or the starting point in the drug design, it is very normal to actually have many side effects for this lead compound, but that's okay because we're looking for pioneering compound to open the door for us, and then we could use that to design better ones, as we'll see in the next phase, in the drug design phase. Okay, so where do we find the lead compounds? This table shows the sources that could be used to find lead compounds. The first one, and actually the most common one, is from natural products. Okay, although currently the other resources are more common, but if you look at the drugs in the market right now, the majority of them have been designed and discovered from natural products. Examples of these natural products could be things like the plant, like flowers, trees, bushes, etc. They could also be microorganisms like bacteria and fungi. Like, for example, the penicillin was actually discovered or released from fungus. Okay. So in that case, it's the source for the penicillin was a microorganism. It can also be found from from animals like frogs, snakes, scorpions. For example, captopril, the blood pressure medication, was actually taken from the venom of snake. Okay, and then you have marine chemistry, so like the corals, fish, bacteria, and I'm not sure why bacteria is listed here with the marine chemistry. It must be a type because there's a bacteria here with the microorganisms. Okay, so let's scratch this. And then there's also a medical folklore, like like historical folklores or traditions that people can um, read and find about new resources. I think I have an example of this later on. And then there is the biochemicals, like in our body, like neurotransmitters and hormones. I mean, obviously, if you know how the neurotransmitters and hormones work in our body, then we could use these as lead compounds, and as a starting point, and then we could modify these compounds to design new drugs. Okay, so um, these are examples of the natural products. Source natural products being used as sources for lead compounds. The next one is the synthetic world. There are certain libraries, and libraries, I mean by saying libraries, I mean like collection of molecules, collection of compounds. There are certain libraries that were designed specifically to be used as resources um, to look for lead compounds. So these libraries were designed like through automated chemical synthesis, and then you could screen these libraries of these synthetic compounds and um, and even the existing drugs to find a lead compound, and hopefully you'll be able to um, maybe um, discover something that has a new um, medical use. I guess my point is, when you look for existing drugs, you actually look for other actions for these drugs. These actions could be a side effect, for example, and then you would enhance that side effect and use it for treating a different disease. Um, I'll give you an example, like in the case of the Benadryl, it's used for allergies, but the side effect for Benadryl is that it causes drowsiness. So now imagine if you are looking for a drug to help somebody fall asleep, then you could use a certain existing drugs like Benadryl that has a side effect that makes people sleep, for example, help you sleep, and then use that side effect as your therapeutic effect. So now the side effect became as the primary pharmacological effect in this case, because you're looking for a drug that would help someone fall asleep. Another example is combinatorial and parallel synthesis, which basically means you have certain chemical reactions that are already predefined. You start with one point and then you explore all the possibilities of designing other combinations of function groups and then end up having a combinatorial library. And the same with the parallel synthesis. You basically start with one function group or one starting material, and then you synthesize others based on the predefined chemical reactions. So the synthetic world actually represents another source where you could dig for in order to find a lead compound. Okay, and then you also have the virtual world, which is basically it's actually similar to the synthetic world, except these are only so these molecules have not been synthesized. They have been like basically designed only on the computer, but they have not been synthesized like in labs. So let's say in the case of the combinatorial and parallel synthesis, instead of actually carrying these chemical reactions to design new molecules, in the case of the virtual world, you would just carry these reactions only in the computer, and then you have a database of these resulting molecules, and you can test these like in silico, like as the in silico test. And then if that turns to be successful and you want to test these molecules, then you could actually go ahead and synthesize them. So you can, as you can see, you can save the time and the money of synthesizing molecules by testing them in the computer first. So this is called the virtual mode. Okay. And the next resource for a lead compound is called fragment-based lead discovery. This is actually a very useful way of designing a drug. I'm going to give you a brief description here and follow that with a figure, but this is just for your information. You don't have to worry about that much. It's just, you know, for your own education. Okay, so basically it relies on the use of NMR spectroscopy to design new drug. Okay, like design something from scratch. Okay, and this method is also called structure activity relationship by, which is SAR by NMR, and you will find, I'll explain why is it called this way when I show this figure for you. So the figure I'm going to show now is just for your education. If you're not interested, you could just skip it. Okay. Okay, so in this figure I'm going to try to explain the fragment-based lead discovery. If this is your protein, and um, if you don't know already, most of the drug targets are actually protein in nature. Okay, so if this is your target drug, if this is your target that you want to design a drug for, and this is the binding site for the drug that you want to design, so then the first step would be that you would screen um, fragments, not full drugs, but fragments like function groups. And that's why this method is called fragment-based lead discovery. So you screen fragments of small molecules, and you would see if any would bind to the binding site, not the whole binding site, but just part of it. And then you would use NMR to see if there's any shift in this binding site. And then if the shift is basically detected, then you would know that there is a binding here. So you would optimize this drug and improve it such that it perfectly fits into the first part, first part of the binding site. You repeat the process again, you find another fragment, you optimize it, and then you link these two together. Once you link these together, then you have a full drug or full molecule that would have high potency because it perfectly fits the binding site. And when I say high potency, I mean the drug has high affinity, high binding interactions with the receptor. Okay, we will talk about the affinity, potency, and all these terms later on. So this is called SAR by NMR because it uses the NMR method, and it also looks at the structure activity relationship. So you add a structure, and then you look at the activity, and you enhance the structure to improve the activity. So this is again just for your information. Okay, so we have talked about the FBLD, fragment-based lead discovery, as a source of lead compounds. Another source would be the mechanism-based drug discovery, and this is also one of my favorite ways of discovering drugs because it relies on computer simulation, and it actually uses the basic fundamental knowledge of the chemistry, physics, and it simulates the receptor, and we also try to find what drugs can bind to this receptor, and then helps you understand. Well, first you understand the mechanism of action, and then you could design drugs that can bind, and these drugs are used as lead compounds. Okay. The last source, or the last example, is serendipity, which means you are discovering drugs by luck. Basically, this is actually, believe it or not, has a lot of a lot of cases where drugs were just discovered by luck. We'll talk about for example, we talked about the penicillin. Penicillin was actually discovered by luck. I encourage you to search for some information, like videos online to hear the story of how penicillin was discovered. It was just basically pure luck, where Alexander Fleming noticed that one of the fungus that he left, actually one of the bacteria peridish that he left for vacation, and came back to find that a fungus grew on that bacteria peridish, and he released what's called penicillin. The penicillin inhibited the growth of the bacteria, but his attention to details and his curiosity has led him to discover the penicillin. But again, it was also by luck. So. And then just a side note, that existing drugs actually can be used as lead compounds, just like I explained here with the synthetic word. We could start with them. We can use their side effect, for example, and design drugs that have better pharmacological activity. Basically, like we said with the Benadryl, the side effect will be enhanced to become your pharmacological or therapeutic effect. Okay, so you could enhance the Benadryl such that it becomes a potent drug for helping someone
sleep. Okay, so basically treating insomnia. Insomnia is the term used for people who cannot sleep. Okay, and then the next step in this phase would be isolation and purification if necessary. And I say if necessary because in the case of the natural world, you would need to isolate and purify the active drug. Like if you have a plant, for example, that has some activity, then you would look into that mixture of compounds and then find which um, molecule is responsible for that activity. Okay, um, and that molecule would be called active principle. So in natural products, the active principle is the compound that's isolated from an extract um, and is responsible for the pharmacological activity. Okay, and of course, the how easy it is to isolate or purify the compound depends on its structure and the stability of that structure and how much quantity do I have of that compound. Like if you have only minor quantity, it would be difficult to extract it among the rest of the other compounds. Example of methods of the isolations and purifications are um, just for your own information, like solvent-solvent extraction, chromatography, crystallization, and distillation. And then the next and last step in this phase is basically the structure determination. So let's say that you found there's a certain molecule in that plant is responsible for the activity and you were able to isolate it. And now we want to find out how the structure looks like. And there are certain methods that could be used, such as mass spec, infrared spectroscopy, ultraviolet spectroscopy, and the most two common ways are actually NMR spectroscopy and X-ray crystallography. The difference between these two is in the case of the X-ray crystallography is just like the chest, just like for example when you take a chest X-ray, so it gives you like a frozen picture of how this molecule is bound to its target, and you could look at the structure and basically decide um, how you can enhance it. With the NMR, you actually look at a solution, so the molecule and the target could be interacting with each other, and this but it can also be performed on solid phases. Okay. Okay, so now that we have a lead compound to start our design process, we could move into the next phase. So like we said, the lead compound usually would have some side effects, and the goal of the next phase is to actually design, improve, optimize actually the lead compound such that it has the least possible side effects and enhance its other features like selectivity, and its maybe pharmacokinetic dynamic properties, as we'll talk here very soon. So in the drug design phase, or also known as the lead optimization, the first step is to identify the structural features that are responsible for the activity. This is known as structure activity relationship, SAR. In fact, this term we will use like a lot, and so just want to make sure anytime we say SAR or SAR, then we should immediately know that we refer to structure activity relationship. In fact, even in NAPLEX exams, they refer to the structure activity relationship by SAR, so you want to make sure that this term is you're familiar with, which I'm sure by the time you graduate you will be familiar with. Okay, so the aim of this stage is, or this step, is basically identifying the function groups that are important for binding. And the method here makes full sense. I mean, so the method of identifying the function groups that are important is basically you alter, remove, or mask a function group. Let's say this OH group, you want to find out if it's important or not. So you would actually change it or remove it or maybe mask it. Mask it like means like you add another one to um, prevent it from binding with the receptor or the target. Okay, and then you would test the drug, and if the activity drops, it means that a function of this group was important for activity, right? Otherwise, um, the activity should not be changed. And then, of course, if the activity is not affected, then it means this group is not important. And then you repeat this for all the groups here, and the ones highlighted here in blue were found to be important. So now you have these structures, you know that they are important for binding with the target, whether it's a receptor or enzyme. Okay, um, this structure is an example of drug called glipine. I believe it's for diabetes. And then, so now you have these structures, and this is called the structure activity relationships. So you have defined the structure activity relationships. Okay. And then the next step is identifying what's called a pharmacophore. So a pharmacophore again are the important function groups, but that's not enough. You want to identify how these function groups are um, um, positioned in relative to each other in space. In other words, you want to define the distances, the angles between these function groups. Okay, because these function groups will be actually interacting with another function groups. Let's say that this is your receptor on this side. So these groups will be interacting with other function groups at the target receptor or enzyme, and therefore the position of these groups is important. Okay, so um, only this. So just by knowing that these function groups are important is not enough. You want to define how these groups are positioned next to each other. Okay, so if this nitrogen is positioned here, for example, then it might not be able to bind to the receptor, and then it's not gonna give you the pharmacological activity required. Okay, by doing so, then you have what's called the pharmacophore, which is basically the 3D feature of the 3D structure for these important groups. So you basically add, um, you look at the 3D component for these groups. Okay, so why do I need to define the structure activity relationship and the pharmacophore? Because once you do that, you could actually start to define these groups by other similar groups, and these similar groups are called bioisosteres. Don't worry about it now, we'll visit it again, but just for your own information, we could modify these groups to enhance these features. First one is the pharmaceutical properties, and then pharmacokinetic properties and pharmacodynamic properties. Let me explain the different um, um, properties here by this figure. So let's say that you take drug orally, and so you administer the drug orally, and then now the drug goes into your stomach, so it has to, the tablet or the capsule has to disintegrate and then dissolve, okay, into the stomach solution or the intestine solution. And in this phase, anything that we do to enhance the dissolution of the drug or the stability of the drug as a tablet, or the dosage form, or maybe the delivery system or stability of the drug, anything we um, work on to enhance these features will be part of the pharmaceutical phase. Okay, so you, the goal at this step is to enhance the administration related properties for the drug, its ability to dissolve, to disintegrate, and maybe if it's you want to give the drug like in an extended release form, for example, then you would work on this phase. Okay, um, and think about the other routes of administration for drugs and what features we can optimize. So in the case of maybe a transdermal patch, for example, right? In the case of a nicotine patch. So think of what parameters you want to optimize to make the batch, for example, maybe last longer, or to improve the penetration of the drug through the skin. All this is considered part of the pharmaceutical phase, and Dr. Gunasilan and Dr. Talukdar would be responsible for explaining all these concepts in the pharmaceutical phase. Okay, and then now after the drug is absorbed, whether it's orally or maybe through the skin transdermally, then it goes into, then we start worrying about another phase, which is called the kinetic phase. So after the drug is in the blood, basically, now it's in the, we worry about the pharmacokinetic phase. In fact, part of that phase is the absorption, which is in this case how the drug is actually crossing the, maybe like the gut wall into the blood, or in the case of the transdermal patch, how the drug crosses the patch to reach into the blood. So now anything that enhances the absorption of the drug is part of the pharmacokinetic phase, and our goal in this phase is to enhance the absorption, making the drug more lipophilic, because the gut wall, for example, is lipophilic. But you want to keep in mind that it should still be soluble in water, so you need to find a fine balance, a good balance between being lipophilic and hydrophilic, as we will visit again later on. So then once the drug is absorbed, it's in the blood circulation, it's gonna be distributed by the blood into the other parts, and at other parts of the body, reaching the target site, and then during the distribution phase, it could maybe bind to some like fatty tissues and accumulate and cause a long, this would give the drug longer duration of action. Or it could be going through the liver, metabolized, and then some of the metabolites could, the resulting metabolites could still be active, for example, so they would go back into distribution. And it could be excreted through the kidney, for example, or excreted the stool, or it could be excreted after it's metabolized. So this phase includes absorption, distribution, metabolism, excretion, shortened for ADME. Okay, so anytime we talk about kinetic phase, then we refer to the four processes, which is called the ADME. Okay, so make sure you catch this term and know that all these steps refer to the pharmacokinetic phase. In fact, this phase can also be referred to as the effect of the body on the drug. Basically, the body is affecting the absorption of the drug, its distribution, metabolism, and excretion. Okay. Now, once the drug is being distributed and it reaches its target receptor, then we're talking about a different phase, which is the pharmacodynamic phase. In the pharmacodynamic phase, we care about how the drug interacts with its target receptor. And if you remember, with the selectivity, we want to have the drug very selective to its target receptor, such that we minimize the toxic effect and enhance the therapeutic effect. This phase is best described as the effect of the drug in the body. Okay, so in the pharmacodynamic, we have the effect of the drug. We worry about the effect of the drug on the body, basically by looking at the interaction between the drug and the receptor, and we try to enhance this interaction by increasing the affinity, the interactions with the receptor, and by enhancing the affinity, we enhance the potency. We could also enhance how efficacious is the drug, and all these terms we will discuss in the future. But for now, the pharmacodynamic phase, I want you to differentiate between pharmacodynamic phase, pharmacokinetics, and pharmaceutical phase. Okay, so and let's see here. Yes, I wanted to say that all these phases are important. You cannot ignore one over the other. For example, if the drug binds very well to the receptor, but it has no way to reach into that receptor, then it will be useless. The same with the pharmaceutical phase. If the drug cannot be dissolved cannot be administered properly, then we will not be able to have a successful drug. Okay, so all these should work together to, make sure we receive the pharmacological effect. So it's like a journey where you start with the administration of the drug, dissolution, and then it's absorbed, and now it's distributed into the body through to all the parts, and then during that distribution it reaches the target receptor, and it causes the therapeutic and hopefully not much of the toxic or side effect, but then it's also metabolized and excreted. So the goal is to enhance this drug, modifying these functional groups and enhance all these features to optimize the drug. Okay, so this concludes the phase of the drug design. So we started with drug discovery. We had a lead, a starting point, a compound that might have some side effect, and then we work on optimizing that drug. Okay, and we'll talk about the next phase now.
The last stage we're going to talk about is the drug development stage, which is actually significantly more expensive than the earlier two stages, the drug discovery and design, mainly because of the clinical studies, because it's at this stage when the clinical trials are performed, and these are performed on a human, so these are the more, and this step is very expensive. In this stage, there are three processes performed in parallel, meaning that you don't have to start like with 1, 2, 3. You could actually start with these three at the same time. The first one is the preclinical and clinical trial. At this stage, you want to make sure the drug is safe and effective by checking its pharmacology, mechanism of action, and test the selectivity of the drug on the desired targets, and if there are any other targets that could be the drug can act on and might cause side effects. Toxicity. So we're gonna at this stage the in vitro and vivo tests are performed to check for acute or chronic toxicity. Metabolism. At this stage, all the drug metabolites are studied and their properties, their toxicity. Basically, we identify these metabolites and study them. And then it's also at this stage when the drug formulations, for example, are we gonna deliver the drug in the tablet form? Which dosage form should be used? What is the best route of administration? And then, like I said, the clinical trials, phase 1, 2, 3, and 4. As you can see, the phase 1 starts with 20 to 100 subjects, and then at phase 2, we have from 100 to 200 patients. Notice here that at phase 1 we're dealing with subjects, not patients. At phase 2 and later we deal with patients, and then so here we check for the pharmacokinetics. At phase 2 we check if the drug works for these patients, and then a double-blind study is performed at phase 3. Notice the increase in the number of patients. And then phase 4 is basically after we market the drug, we keep track of the side effects and how the drug is performing. Notice that this is the number of years. It takes about from 8 to 9 years to discover a drug. The patent expires 20 years after filing for the application, which is one of the steps here, filing for the application and following the regulations by the FDA and the GMP and GCP. Also, one of these steps in the drug development is designing efficient manufacturing technique, efficient synthesizing technique for large quantities. This is an important step. In fact, organic chemists compete to find the most efficient and fastest way to develop large quantities of the drug, and cheapest way, of course. And of course, at this stage also is when the drug is registered, marketed, and sold to make money.